Pablo es ingeniero bioquímico de la Universidad de Mendoza, después hizo un doctorado en química en la UBA y nos encontramos acá en los pasillos de CONEA cuando hizo su postdoctorado en nanopartículas de sílica para aplicaciones biomédicas y después siguió su camino en Estados Unidos, en La Joya, donde estudió el microambiente tumoral. Después se fue a Estonia, donde cayó en un laboratorio pionero de péptidos antitumorales y siguió ahí varios años y hoy en día es un investigador del CSIC, que sería nuestro CONICET España. Tiene un montón de estudios en péptidos, tiene patentes, pero bueno, sobre todo más allá de esta brillante carrera académica, es una persona maravillosa. Compartimos muchas frustraciones de Ferrari en Fórmula 1 y terminar los viajes en 140 después de todo un día acá haciendo experimentos. Así que es para mí un placer presentarlo y que haya aceptado esta charla.
Bueno, muchísimas gracias, Marina. Gracias a todos a los organizadores, bueno, Eve, Marina, Alexis también. Y bueno, si no les molesta, voy a hacer un switch a inglés porque la tenía preparada en inglés. Okay, so today I will present about new targets and targeting peptides for glioblastoma. Let me change this. And you can see the slides, right? Okay, correct me if not. This is our research team, our current research team. We are located in Barcelona, near the downtown of Barcelona. And as Marina mentioned, it's an institute that belongs to CSIC, which is the National Research Council of Spain. And there are several institutes in Spain and also in Catalonia. This one is the Institute for Advanced Chemistry of Catalonia. And let me talk to you a bit first about the work that we do in our lab. We try to develop targeted therapies, and we call them peptide-guided therapies because they're guided by peptides. So the first thing we do is we like to probe a disease that can be glioblastoma, but also other diseases, in vivo with a peptide library. And this technology is called in vivo peptide phage display. And this gives us a targeting peptide. And with this targeting peptide, we do two things. First, with it, we develop guided drug delivery systems. And then also, we identify what the peptide binds to, so its receptor. And this is very important because it gives us information about molecular changes that occur in the disease. And by doing this, we normally end up identifying new targets and biomarkers. So why do we like to work with peptides? Peptides are the way that nature uses to recognize two proteins. The binding between two proteins happens not using the whole surface of the protein, but tiny bits of it that are peptides. And these peptides can be as short as 3 amino acids. And that was shown already in 1984 by Roslati and Pierce-Bacher in this Nature paper, where they showed that the interaction between integrin and fibronectin occurs between integrin and the tripeptidic RGD motif of fibronectin. So this means that we can use peptides as targeting elements to guide drug delivery. But also if the peptide binds to an active site, it could have activity on its own. There are several ways to identify peptides. You can do screening with a library of peptides, which is what we do. And you can also identify them computationally. And there are several advantages for drug delivery of peptides. They have a fast excretion rate, which is good for radiotherapy because you don't want to have a very long circulating radioactive agent in your blood. They can be modified to make them orally available. And they are normally less immunogenic than monoclonal antibodies, and they have high penetration in solid tumors, and you can scale their production at GMP. Now, the affinity of these peptides, these short targeting peptides, is normally quite lower than the one of antibodies, and this can be a good thing because it has been shown that for antibodies, if you have too high affinity, this can preclude the penetration in tumors. And this was shown here for this study in tumor mice, where you can see that the antibody drops in penetration when you increase the affinity towards a picomolar range. And it was shown also for human tumors. Here you can see in green the penetration of this antibody against EGFR that has an affinity of 50 picomolar. And you can see that basically the antibody remained only in the border, in spite of the fact that the core of the tumor has the receptor. Peptides are a big chunk of the pharma industry. There's already 80 peptide-based drugs that are approved. Last year, the second most sold drug in the market was a peptide, the famous Ozempic peptide. And most of the other ones that are approved are hormonal analogs like insulin or oxytocin. And they are also antibacterial and antifungal peptides. And most of these are cyclic. For cancer, we also have several that are approved, like lurbinectedin, motixafortide, and octreotide, which is an analog of somatostatin, and it is used in the radiotherapy called Lutathera. This is a peptide that is a radioligand of lutetium-177, and it's used for radiotherapy. So we identify targeting peptides by doing in vivo peptide phage display. This was a technique that was pioneered by Roslatti and Pasqualini in 1996 in a Nature paper in the lab where I later performed my postdoc studies. And here you construct a library of peptides by expressing random peptides on the code surface, on the code protein of a certain virus. This virus is a bacteriophage, means it infects the bacteria, and we refer to it simply as phage. In this case, the peptide insert that we use has 7 random amino acids and 2 cysteines in the end, and this gives us a diversity of library of 10 to the 9 different clones. So if you want to find the targeting peptide for a tumor, for example, let's say this is a tumor, you inject the library intravenously in the mouse. After 30 minutes, you perfuse. This will get rid of all the phage that are in the circulation and the one that are loosely bound. And then you sacrifice the mouse and you collect the tumor. And then the phage that are there are still infective. So after you collect the tumor, you can rescue the phage by doing a homogenate of this tumor, and you basically mix with the bacteria, which is the host. The phage get amplified, and this is a very simple way also to purify the phage from the rest of the tissue. So once you have the purified phage, then you can sequence the genomic DNA, and this will have the information of which were the peptides of the clones that bound to your tumor. And in our case, once we have this information of the peptidic sequence, we simply work with the synthetic peptide. So this technique is nice because it interrogates the protein in the right environment. It means the correct conformation it has in the disease. The first step is in vivo, so the peptide you obtain will be able to penetrate the barriers, and it probes mainly the vasculature. And we know that the different diseases like cancer, they put a molecular signature on the vasculature very early on. So by doing this, you can detect early disease biomarkers. Also, because the phage is a biological nanoparticle, then the peptide, if you take this peptide and put it on a synthetic nanoparticle, it will most likely have the same targeting effect. So now I will describe the targeting peptides that we identified in the last years for glioblastoma. And interestingly, these studies started not from studying glioblastoma, but by studying Alzheimer's disease. And they started already a while back. I was still in La Jolla, and we were interested in finding peptides that would target the vasculature of Alzheimer's disease in mice. So we performed this in vivo phage display. We injected the phage library in Alzheimer's disease mice and also in healthy mice. And after 30 minutes, we collected the hippocampus, and then we sequenced the rescued phage. And we got several candidate peptides. One of those candidate peptides we call DAG. You can see here the sequence. And this was the first peptide that we tested, and we already published it several years back in 2017 in this Nature Communications paper. And there we showed that the peptide did target Alzheimer's disease, but we also treated in glioblastoma. And we saw that it homed very well to a patient-derived xenograft of angiogenic glioblastoma. And it did so by binding to the protein connective tissue growth factor, which is a matricellular protein, and it's secreted by activated astrocytes. So this is already published, but then in 2018, some years later, I became more interested in glioblastoma. So I decided to test another peptide from this same screen that we refer to as CES. And the sequence is here. It's also a cyclic 9 amino acid peptide. So the first thing we did was synthesize the CES peptide, and we coupled to it a fluorescein probe, and we injected this peptide intravenously in mice with UAT7 glioblastoma. And 1 hour later, we perfused the mice and we checked the tissues with confocal microscopy. And you can see here in green is the signal from the peptide. This peptide accumulated very well in the glioblastoma, and it showed some colocalization with the vasculature, as you can see here in yellow. We then tested this peptide in another model of glioblastoma, which is referred to as the wild-type GBN model. This was developed by the Gabriel Berger's lab in San Francisco in 2013, and it's derived from hippocampal astrocytes from neonatal mice that are modified with 2 oncogenes. And these cells, if you inject them in the brain, they develop very aggressive tumors. We saw very similar trend here that our CES peptide accumulated in glioblastoma and also showed colocalization with CD31. So the most interesting part of this in vivo phage display technology normally comes when you identify the receptor, so what your peptide binds to. And in this case, we identified the receptor using magnetic beads modified with the CES peptide, and we did a pull-down experiment using UAT7 tumors, and proteomics gave us a candidate receptor, this protein SNAP25. This is called synaptosomal associated protein, 25 kilodalton, and it's a protein that forms a complex with other proteins of the snare complex, and its function is to fuse the membrane of synaptic or secretory vesicles with the plasma membrane of the cells, so that the content inside the vesicles can be released, such as neurotransmitters or hormones. And it is normally accepted as only intracellular protein. So the first thing we did was validate or check if the peptide CES binds to recombinant SNAP25. And we showed this by ELISA and also by quartz crystal microbalance, which showed that the affinity of CES for SNAP25 is 25 nanomolar. And this is exceptionally good for these short targeting peptides. We also checked our tissue sections of glioblastoma injected with the CES peptide, and we saw that the CES peptide was colocalizing with SNAP25 in these glioblastoma sections. Okay, so as I mentioned, the SNAP25 is accepted to be an intracellular protein, and normally our targeting peptides do not bind to intracellular targets. They bind to something on the vasculature or on the cell membrane. So we hypothesized that if CES is binding to SNAP, then perhaps in glioblastoma something is going on, and this SNAP25 is going to the cell surface, and it's becoming accessible to peptides or to antibodies. So we checked this with live cell flow cytometry without any detergent, and we could see a clear SNAP25 positive population in the U87 cells. We also observed very atypical staining pattern in our glioblastoma mice. You can see here we observed these SNAP25 positive deposits on the blood vessels of glioblastoma. And then we did an experiment to see the accessibility of SNAP25 from the circulation. So we administered an anti-SNAP25 antibody in glioblastoma mice, and we saw basically where the antibody ended up. And we saw that it only accumulated in the tumor, but not in any of the other organs. So this is a strong suggestion that SNAP25 is becoming accessible only in the tumor, which makes it a really good target for drug delivery. So then, secondly, we hypothesized that if this protein is on the surface of the cell in glioblastoma, perhaps it's also interacting with the extracellular matrix. So we checked if recombinant SNAP25 binds to ECM proteins using an array of ECM proteins, and we saw that SNAP25 mainly bound to collagen 6. And this finding was further corroborated using surface plasmon resonance, and this showed that SNAP25 binds to collagen 6 with a quite high affinity of 15 nanomolar. Another observation in vitro was that the U87 cells were shedding what appeared to be microvesicles, and they were positive for SNAP25. Also, the cells adhere to collagen 6 surfaces. You can see here, much more than to plastic. And this adhesion was abolished with an anti-SNAP25 antibody or with a free CES peptide. So then for drug delivery applications, we designed a peptide drug conjugate of CES for photodynamic therapy. So we took the CES peptide, shown here in red, and we coupled it via a linker to this molecule that is called verteporfin. Verteporfin is a small molecule. It's used as a photosensitizer for photodynamic therapy because when you irradiate it at 690 nanometers, it produces reactive oxygen species. The nice thing about this molecule is that it's FDA approved, at least for ophthalmology. But one caveat is that it is not water soluble. However, our conjugate not only targets the verteporfin to a specific site, but it also solubilizes it because this is water soluble. We checked the stability of both CES peptide and the conjugate in serum, and they showed to be fairly stable up to 48 hours. And then we performed in vitro photodynamic therapy experiments, and we could see that our CES-V conjugate was selectively toxic to these cells that express glioblastoma cells that express SNAP25, whereas the control conjugate was not. And also, the CES-V conjugate was not toxic to cells that were negative for our receptor. So now we're looking into the SNAP25 expression in human glioblastoma samples. And this is a collaboration so far with the Tartu University Hospital, who donated 10 glioblastoma patient samples. And here we did immunohistochemistry for SNAP and other markers in the absence of permeabilization. And what we observed first is that there is an overall loss in SNAP25. Here you can see the quantification of the SNAP25 intensity in the whole brain. This is the value for healthy brain. And these are the different patients. So normally there's a loss of SNAP25. And however, here you can see representative images. This is a healthy brain. There are regions that highly express the SNAP25, like this one. And also there are regions that show a complete loss of the SNAP25, like this one. We also observed very atypical patterns and localization for SNAP25. Here I'm showing an image that is representative. Well, it shows that these tissues, these tumors are fully loaded with these structures that you see here that appear as vesicles, and they also appear to be extracellular. And we measure them, and they are approximately between 1 and 3 microns, the same size that we see for the U87 cells in vitro. And we also detect SNAP25 presence on the blood vessels. And these blood vessels that have SNAP25 also appear to be positive for collagen 6. Next, we checked this new marker, the SNAP25. We looked it up in this curated cancer cell atlas, which is an atlas that contains single-cell RNA-seq data for 1,000 human tumors. And indeed, it was high and present in brain, but it was even higher in neuroendocrine tumors. So based on this, we decided to study it in neuroendocrine tumors. So we took a couple of cell lines, neuroendocrine cancer cell lines. One is rat pancreatic and the other is mouse gastric. And we, with flow cytometry, showed that these indeed express also superficial SNAP25. So these are ongoing studies. For conclusions, take-home messages is that we developed 2 targeting peptides for glioblastoma. The DAG targeting peptide targets glioblastoma by binding to CTGF. The CES peptide targets glioblastoma by binding to SNAP25 with high affinity. And it can be used to deliver a photosensitizer. And SNAP25, we believe we have identified possibly a new target in glioblastoma. It was very specific to glioblastoma, not because of its expression levels, but because of its differential accessibility in the tumor. And it showed very atypical expression first on the cell surface, but also on extracellular vesicles and on the blood vessels. And also importantly, it binds to collagen 6. So now we're performing different studies to try to understand what is going on with this, what is the role of SNAP25 and also the role of the vesicles. But moving forward, and these are also ongoing steps, we want to use our CES peptide as a radioligand for radiotherapy. And this is an ongoing collaboration with the CONEA in Buenos Aires, with Lisa Tomás, López Bularte, Marina Perone, and Oscar Pozzi. And we're radiolabeling the CES via a DOTA with the lutetium-177, which is a beta emitter. And it's already used with success in at least for neuroendocrine tumors with the radioligand that I showed you before, Lutathera, which is a peptide that is very similar to our peptide. It's similar in structure and also similar in size, only that it binds a different target. So we believe that CES has a clinical potential. We're also studying the palmitoylation inhibition in glioblastoma because SNAP25 is normally palmitoylated. So we're looking into maybe palmitoylation defects that may explain the location of SNAP25 that we see. We are planning to do in vivo photodynamic therapy with the CES verteporfin conjugate, in combination also with microglial reprogramming, and this is an intense area of study of our lab. We recently developed the macrophage reprogramming agent called MacType-V. And I want to thank my funding agencies, and most of the work that I showed was done by these two people. Alberto Guillermo Arias is an MD. He is finishing his PhD in CONEA. He's now a visiting researcher here in Barcelona. And Laura Tovar Martinez is a PhD candidate. And these are all my current collaborators. And thanks again. I'm happy to take questions.
Muchas gracias, Pablo. Excelente charla. ¿Preguntas? Y acá está Maru para preguntar. Hola. Hola, Pablo. Bueno, no
I
ves, pero
cannot
estamos acá.
hear you.
¿Me escuchas? ¿Sí? Pablo, ¿nos escuchás?
No, no escucho.
Hola. Sí.
¿A mí me escuchás, Pablo?
Yo no escucho.
Hola. ¿Ahora nos escuchás? No.
¿Me escuchás a mí, Pablo?
Hola, hola. Ah, como que la gente escucha. Pero de allá Pablo no está escuchando.
Qué raro, yo no escucho nada.
Si no, le pongo las preguntas en el chat. No sé qué opinan.
¿Ustedes sí me escuchan?
Yo escucho todo.
Blanca 10 escucha. Hola, doctora 10.
¿Cómo va? ¿Todo bien?
Bien, por suerte. Gracias
Yo
por
los
venir.
escucho a todos, o sea, a los que no escuchan y a los que dicen que están ahí, así que no
Por
sé
ahí
cuál.
si mutean y vuelven a encenderlo.
No, hay que compartir su pantalla.
No, pero es que no me escucha a mí. No escucha a nadie, Pablo, me parece que tampoco la escuchó a Blanca. Ah, ¿le escribís, Caro, ahí? Que capaz deje de compartir. Vaya, dejó de compartir.
Yo le escribí en el chat, pero
Que salga y entre.
él no nos escucha a nosotros. No sé si Mari, si vos le podés decir por WhatsApp.
Yo ya me olvidé la pregunta.
¿Eh?
No, la tengo anotada,
Ahora sí, ahora escucho.
Ay, buenísimo, buenísimo. Hola, Pablo. Vos no nos ves, pero acá somos un montón. Muy buena tu charla, muy interesante el trabajo.
Gracias.
Yo tengo una pregunta. Vos tenés, ahí ves que el SNAP 25 hace shedding con estas vesículas. Si vos querés, bueno, para terapia fotosensibilizadora o para radioterapia, no importa que tu péptido esté pegado a las vesículas porque se queda ahí en la parte del tumor, ¿no?
Sí, sí.
Porque ahí ese shedding, como que si quisieras, por ejemplo, el péptido, ¿podrías combinarlo con un quimioterapéutico?
A ver, no estamos seguros sobre el destino celular del péptido dentro de la célula, no estamos seguros dónde termina. Vemos que por los resultados de terapia fotodinámica in vitro, vemos que es bien tóxico selectivamente, pero no estamos del todo seguros si entra a la célula, porque ese efecto podría darse simplemente si el péptido se queda pegado a la membrana, pero bueno, eso lo estamos mirando y si el péptido entra a la célula, perfectamente se podría acoplar al péptido, en vez del sensibilizador, se podría acoplar un fármaco.
Porque lo interesante de acoplarlo a un fármaco quimioterapéutico es como que podría ser más rápida la parte translacional de esa estrategia, ¿no? Pero igualmente,
Sí,
si vos, por más que se pega a las vesículas, lo podés acumular el quimioterapéutico en la zona del tumor.
sí, sí, exacto, exactamente.
Serviría igual, ¿no?
Sí, pero bueno, igual es algo que sí o sí tenemos que saber, porque si uno quiere llevar, por ejemplo, como fármaco, si querés llevar sirna o eso,
Ahí
entonces sí
sí,
es importante que entre a la célula.
ahí tenés que entrarlo a la célula, claro.
Sí, pero bueno, también en lo de la vía fotodinámica lo queremos hacer también como prueba de concepto, pero lo que más nos entusiasma es la idea de usar el péptido para radioterapia, para llevar
Claro,
lutecios.
sí. Ahí te pregunto algo re cortito. ¿Estudiaron si hay vesículas con SNAP 25 circulantes como para ver
No,
seguridad de la estrategia?
sí, esa es muy buena pregunta. De hecho, lo queremos estudiar, estamos viendo a ver si tenemos acceso a alguna seroterapia. Porque yo creo que puede llegar a haber, porque en glioblastoma no está reportado, pero sí está reportado en Alzheimer, que hay presencia de vesículas que son positivas a SNAP 25 en el suero. Y bueno, eso sería interesante ver si está. Podría ser algo bueno, porque tal vez sirve como agente pronóstico, pero también, como vos decís, si hay muchas, tal vez podría ser algo malo porque nuestro agente se une a lo que está en la sangre y eso no querríamos.
Claro, sería cuestión de evaluarlo. No
Sí.
quiero, tengo un montón más, pero hay personas preguntando en el chat. Estaba Blanca 10 y alguien más. ¿Le damos a Blanca 10 la palabra? Blanca, pregunta si querés.
Sí, lo que yo quería saber si estas vesículas extracelulares, además del SNAP, tienen otros marcadores, como por ejemplo el receptor de crecimiento epidérmico, que se sabe que en glioblastoma forma vesículas o anillos extra membrana, digamos.
Sí, sí. Sí, muy bueno. Nosotros teníamos la misma sospecha y de hecho ahora estamos tratando de aislar las vesículas. No es del todo fácil aislar vesículas, pero nuestro plan es aislar las vesículas de cultivo y después hacerle proteómica, también ver el contenido de Mirnas, porque está eso que decís vos del el VGF. Y además se ha visto que estas vesículas pueden destruir la permeabilidad vascular, rompen la barrera hematoencefálica, así que es muy interesante ver su efecto. Sí, todavía no sabemos, pero nuestro plan es hacer proteómica de esas vesículas.
Gracias.
La-
Nicole, ¿quieres preguntar?
Hola, ¿qué tal? Muy buenos días para todos. Me encantó tu charla, Pablo. La verdad que no
Gracias.
conocía de esta parte. Me parece súper interesante la generación de este tipo de péptidos contra distintos targets. Me parece que es la tecnología que se viene pensando en este diagnóstico personalizado y particularmente interesante, ¿no? En el tema de tratamiento de gliomas pediátricos, que ya se ha visto que tienen un comportamiento además totalmente distinto a los gliomas de adultos, ¿no? Me parece que está muy bueno en ese sentido. Te pregunto, digo, podemos ahondar en esto horas, me parece sumamente interesante, pero te pregunto algo particular porque sinceramente no conocía de SNAP 25. ¿Qué se sabe de la regulación post-transduccional en cuanto a lo que es la ubiquitinación, fosforilación o clivaje proteolítico en tumores? Porque he visto que hay, o sea, hay algo de relación, algo no, una relación muy importante en las cuestiones post-traduccionales y eso afecta mucho también pensando en el rol según el grado y el subtipo molecular de glioma. ¿Ustedes han dado en ese tema?
No lo hemos mirado, no lo hemos investigado, tampoco he visto que esté reportado. Lo que, al menos yo en lo que más estuve leyendo es me centré más en la palmitoilación, pero después en otros aspectos, en glicosilación o otros aspectos o cambios post-translacionales, la verdad que no sabría decirte. Lo
Bien,
único que
porque
sabemos es que el péptido se une a la proteína recombinante que tenemos, está hecha producida por bacterias, entonces no se espera que esté palmitoilada, entonces sabemos que el péptido se une a la proteína no palmitoilada. Bueno, y a la palmitoilada también, pero no necesita el palmítico para la unión.
bien. Bien,
Sí.
porque ahí hay un blanco interesante.
¿Cómo?
Y bueno, que hay un blanco interesante, digo, que está bueno y me parece muy interesante. Así que, nada, gracias por transmitirme un poco lo que tenés experiencia en ese sentido.
Dale, gracias por la pregunta. Lo vamos a estudiar.